So the last one for today, we're going to be doing RNA-Seq. So if you go under transcriptomics, it should be right there. This is definitely one of the more complicated services that we have. So as before, if we go into BV-BRC workshop, AMR workshop, we're down to RNA-Seq. You can see that there are two here, but you should use RNA-Seq demo. That's the more recent one that's had some of the improvements to the pipeline. Because it's a little difficult to cover all the use cases for transcriptomics and the difficulties that arise, this is a service that's constantly being improved. So in its current state, though, we have some good stuff with it. I'll first go over the interface, and then we'll go through and do an example. So the first thing for the RNA-Seq interface is that you are going to be selecting a strategy. So we have three options currently. One uses more state-of-the-art tools, HT-Seq and DE-Seq2. HT-Seq is one of the more popular up-to-date abundance estimation programs, and DE-Seq2 is the state-of-the-art differential expression software. So host HiSat2 is a pipeline made specifically for host organisms. So the only real difference between this and the HT-Seq pipeline is this one uses Bowtie2, which we use to align against bacterial genomes, and HiSat2 is used specifically to align against hosts because of introns and extrons and all that stuff. Tuxedo is a pipeline that relies on cufflinks and cuffdiff. It's not a maintained tool, but we keep it because it has its own advantages. Namely that if you want to do differential expression, and you have to have a minimum of 2 samples per condition if you want to use DE-Seq2. So if you just don't have that bandwidth in your lab and you only have 1 sample for a condition, then you're going to have to use Tuxedo, and it will give you those approximate abundance estimations. So after selecting a strategy, you can go ahead and select your target genome, like with all the other services. There is a trimming option. So if you have raw reads, you can just run those through without trimming them first, and we'll save the trimming output. Just know that depending on the size of your files, this could just take a lot longer. For our example, we'll be selecting false. So make sure you don't submit with true enabled. Output folder, output name. So when you're going through this, and say I'm going to be doing this off of the assumption that you're going to be doing differential expression. So the first thing you want to do before you add any of your samples to the selected libraries is you want to turn on the groups and conditions box right here. And so here you can just put in any arbitrary condition name. I'll just do condition 1. You add it. And then condition 2, you can add it. And then over here in the paired read libraries, you can go through, select. Let's see, I'm going to go back to that example in my workshop. Over here in the reads, I'm going to be selecting some of these sample files. So note that when I select it, select a read in read file 1, and it will be the same for the read file right here. It populates the sample identifier field. So you can go ahead and modify this to whatever name you feel captures the information or whatever experiment was done on that file. So go ahead, select the second one. All right, so since that's all set up, now before we add it to the selected libraries, we have to assign it its condition and then click it over. So I'm going to do another example with 2 more. Here we go. 2. All right, and so this will be the first set for condition 1. So as I said, if we wanted to do this strategy, we have to have 2 samples per replicate. So I'm just going to choose 2 more. And I'm going to make sure to change this to condition 2 before adding it to the library. Stays on condition 2. We add it. And now we have a valid set of reads with assigned conditions in order to run RNA-Seq with differential expression. But the other very important part before you submit your job, well, you won't be able to until you fill out the contrast table down here. And the contrast table is important because if you're going to do differential expression, you have to decide which condition is going to be upregulated and which condition is downregulated. So you're essentially deciding the positive and negative values for your log fold change results. So for this table, when you select condition 1 here and condition 2, this essentially means when we do the differential expression calculation, Positive log fold change values mean it's upregulated in condition 1, and negative log fold change values means it's upregulated in condition 2. And so that's filled out. You have to put in the target genome. Right, this is not, there we go. Okay, so this is what a correct RNA-seq interface form looks like. We have the conditions put in, we have the read set up with the appropriate conditions, and we have the contrast table set up. And so once all that's filled out, you can go ahead and press submit like with every other job, and it will show up with that same message once it has been successfully submitted. And depending on the size of your files, RNA-seq can actually take a very long time, so you will have to be patient. It could take days depending on how big your files are. I think the longest I've had was 2 or 3 days for one of my examples. So we're going to go ahead and go through some of the output. If you go back to the RNA-seq folder, let's go into the Haemophilus RNA-seq demo example. So there's a lot of output here, and I'm just going to start from the top and work my way down. So in this pre-made example, the conditions that were set up were 6-hour infection, 72-hour infection, and the control. So whenever you set up we'll go back to the page. Whenever you set up a condition here, the name of that condition ends up becoming a folder name in your output results. So for example, if we go into 6-hour infection, we can see 2 more folders with the names of our samples. And going into there, you will find all the specific files for that sample with all the output from the tools that were run on it. So you have the BAM file, you have some quality assessments like SAMSTAT and FastQC. If you enabled trimming, this is also going to be where you find the trimmed reads file, as well as a trimming report, etc. You go back. There we go. If you do differential expression, you will see, assuming that it completed with no issues, you will see a file that has the suffix deseq2.tsv. If we click view on that, this will take us to a table here that is all of the differential expression results output from deseq2 or CUFDIF, if that's what you used. Just going through these columns really quickly. Of course, we have the gene name, all of these, the base mean, I guess, the mean value. Log2 fold change, obviously, is the differential expression calculation. Positive, of course, upregulated condition 1, negative upregulated in condition 2. The important part, though, when you're looking at this table is if you are going to look at p-values, you have to look at the adjusted p-value column. Because since differential expression is just a lot of hypothesis tests, you have to adjust your p-values for all of the tests that you're doing. So yeah, we have more of these deseq2 files. I will come back to this other job results. So a few of our services like comprehensive genome analysis do have job results within your job results. And so this one is essentially lets you access a viewer for your differential expression results. And we'll go into that in a minute. So when you get your output, one of the best files to start with is just this BV-BRC RNA-Seq report. And this is just a very bare bones file that has, a brief summary of what you did, a summary table that includes the alignment score for your sample. If you have a job that failed because one of your samples has a very poor alignment, then that will show up in this report and flag the sample that did not align well to your reference genome. And then a list of the references for the programs that were used. Additionally, there's a link to the multi-QC report, which is third-party software that aggregates the outputs from multiple other bioinformatics programs. It's been very useful. So if we open that in a new tab, this will just open up the report that's in the same folder. So this should give you more in-depth information as to the percentage of reads that actually mapped for that sample. And it shows it in a number of different ways. This is your general statistics table. But if we go down, we see the outputs from other programs that were run, like RCXC. Here, we're just trying to predict the straightness of your sample, which is unnecessary for some researchers, but others really like it. So the output from HT-seq count, you can see that there's different categories of the alignments. So these, for example, the blue here means that that gene was assigned uniquely to the reference genome's annotation. This section means that the read assigned ambiguously. Orange means that that read mapped to the reference genome, but there was no feature at that location in the annotation file. And then this purple section, too low quality, generally means that it was multi-mapping, essentially. There were too many locations where that read could map on the reference genome, and so we couldn't assign it concretely to a particular feature. So yeah, this report has a lot of good stuff to analyze your results. Would recommend taking a look at it. We go back to the example. Let's say we have the control, gene counts, matrix, TSV. Like the DE-Seq table, this is just going to be using the TSV viewer. You can just see the raw abundance estimation output from DE-Seq2. Select that. Each of these will have, for example, this sample had 690 hits for this gene, etc. So this is just the table, so you can see all of those gene count results tabulated. This is a supporting file that has summary stats on the differential expression results. So this is just another raw text file. Each of these columns is a differential expression result for each of the comparisons. And this file is important for the differential expression viewer that we'll go over in a minute. There again, there's multi-QC. These mapping files, so actually I'll just go into here. These are used to help with creating some of the images output for the report. So whenever you run RNA-Seq, two things that we do for your samples are try to assign those genes to the pathways and subsystems that we have within BV-BRC and then show them in violin plots. So essentially for each of your conditions in those samples, we try to show the distribution of your gene counts, but here we don't use the raw counts, we use TPM values, which is a good method for comparing the amount of a particular gene between conditions. So it's not so good between sample by sample, but when you have multiple samples that assign to a particular condition, this is a good normalization metric for comparing between those conditions. And so this is an example of the pathways file. We also have the subsystems file. And then if you did differential expression, then we also output volcano plots from the DE-Seq2 results. And the comparison that was done for that one is over here in the top left above the figure. It's important to take a look at these values when you're looking at the volcano plot. The red dots indicate genes that are significant and have a high log2 fold change. So they are something that is significantly different from one condition to another. And so you have these different categories like not significant. The log2 fold change value might be high, but the test was not significant. So you don't want to include that gene in your analysis. And then less common, you have ones that might be considered statistically different, but not at a value that's actually usable for your results. Let's go back. So that finishes report images. The last file in this list up here is this TPM counts matrix, similar to the gene counts matrix and the DE-Seq2 results. This is just a matrix showing the raw TPM values that were used when creating those violin plots that we just saw. So this just shows all that information for each of the genes and your samples. We'll first go into this one. So we see this Homophilous RNA-Seq demo diff.exp file right here. Clicking into that, you can see that there are 4 JSON files here. You really don't want to try to view those. All you want to do is click the experiment button, and this will take you to the differential expression viewer. This works very similarly to the protein family sorter, and that we have the genes here and we have some other information, for instance, the number of comparisons that that gene was involved in and whether or not it was upregulated or downregulated in those comparisons. We have the standard filters down here, so you can filter by log ratio, but this is also absolute value. So if you filter, for instance, by a log ratio of 2, that means you will get both those log ratios that are 2 and greater and -2 and smaller. So we're just going to do that. We'll filter, actually, to show the contrast. So you can see we have 1,741 results before applying the log fold change filter. And after that, we only have 329. So if we go over to the heat map, there you can see like the protein family sorter. We have the genes up here. We have the comparison here. So this is on the x-axis, the contrast. And then you can, as before, apply clustering and find genes that are, upregulated together across different comparisons or downregulated. We've been told to fix this in the past. It's something we intend on doing, but here red means upregulated and green means downregulated in your contrasts. Some people find that should be the opposite way. We might get to changing that, but just keep that in mind when you're looking at this heat map.
Can I select things from that heat map?
You can, just like before. If I wanted to look at this group right there, it'll grab those features. We can add it to a feature group, or we can just go ahead and look at the table and show genes, etc. All the other cool functionality we've been going over in this workshop. All right, and the last piece of functionality I want to show you for RNA-Seq. So one of the common ones is you also want to be able to view the BAM files that are output from RNA-Seq, and you can do that by clicking the browser button here in the top corner. This will essentially go and track down all the BAM files in your subfolders here and take you over to the genome browser. I'm going to have to zoom in pretty far in order to show it. If you ever try to use the BAM file viewer and you click on one of these and it says something like too much data, then just keep zooming in, because it just means that it doesn't have the resolution to display the reads lineup at how far away you are. So here we can see, between these two examples, the stacking of the reads against the reference genome here. And you can just scroll along it. I think that about covers it for RNA-Seq.